the Subcommittee on Healthcare Benefits and Administrative Rules and the Subcommittee on Government Operations will come to order without objection. The chair is authorized to declare a recess at any time, and just for our situational awareness, they're going to call votes around 10:40 is what we understand, and so what we will do is hopefully get through all the opening statements, your opening testimony. We'll recess briefly to go vote, then come back and actually do the Q&A portion of that. Hopefully that works with all of your schedules, but I want to thank all of you for being here. Obviously, the use of fetal tissue in research is something that has become really in the headlines again, most recently because of some of the potential procurement by the federal government here. And it was raised to the awareness of Chairman Jordan and I in terms of an ongoing practice of procurement here at the federal agency. And yet, what we believe needs to make sure gets addressed today is sound science, along with a moral code that, quite frankly, most Americans believe and support. And so I'm looking forward to hearing from all three of you in terms of the science and what's available out there, and yet making sure that what we do as a nation is one that is responsible, one that is truly based on what is available to us today. There's unbelievable technology. I hope that we hear from all three of you based on your statements. I believe that we will hear. But yet there's also a lot of falsehoods that are out there on what this will do or won't do. And I can tell you, as someone who is proudly pro-life and believes in the sanctity of life, it is critically important that we look at all of these issues. And so I look forward to hearing from all three of you. I look forward to this hearing and hopefully drawing attention to what we should be doing as a nation, what we should be doing as a federal government. And with that, I will yield to the ranking member, my good friend, for his opening statement.
Thank you, Mr. Chairman. And thank you to all the witnesses for sharing their testimony with us today. I come to this hearing from the perspective of improving the practice of medicine. The International Society for Stem Cell Research is headquartered in my home state of Illinois. It is the world's leading professional organization of stem cell scientists and represents more than 4,000 members. ISSCR calls fetal tissue, quote unquote, an essential gold standard resource in biomedical research. For more than 80 years, scientists have relied upon the unique qualities of fetal tissue cells, which are able to adapt to new environments in ways that adult cells are not, to make life-saving advances in modern medicine and fight diseases that ravage communities around the world. Fetal tissue research has helped contribute to vaccines for polio, chickenpox, rubella, and shingles, medical achievements responsible for saving thousands of lives. This life-altering research continues today. Fetal cells, called trophoblasts, are critical in efforts to better understand and develop a vaccine for Zika, which causes birth defects to babies in utero. The Centers for Disease Control, the CDC, projects that the number of Americans suffering from Alzheimer's disease will triple by 2060, leaving 14 million Americans suffering from degenerative memory loss and cognitive issues. But promising therapies are being developed using fetal nerve cells to restore damaged neural circuits in the brain. These efforts have the potential to improve the quality of life for millions of seniors and their families, as well as significantly lowering healthcare costs. While there are promising developments in adult stem cell therapies, this early stage research is not yet ready to fully replace medical research that has been effectively used since the 1930s. Rather than being discarded, fetal tissue can push scientific innovation that saves lives. The use of fetal tissue in research is protected by law. The Department of Health and Human Services has set stringent ethical and legal guidelines dictating proper consent, transportation, and reimbursement for all donations from hospitals and clinics, as well as research methods in biomedical facilities that wish to use fetal tissue in their research. The federal government has funded beneficial medical research that uses fetal tissue since the 1950s. Thanks to that support, medical scientists continue to apply the unique and irreplaceable qualities of fetal tissue to discover new ways to save lives and improve the quality of life for millions of Americans and people around the world. Thank you, Mr. Chairman, and I wanted to just bring up one issue. In recent days, the committee has received numerous letters from nonprofit and advocacy organizations, including the Michael J. Fox Foundation, the National Multiple Sclerosis Society, the HIV Medicine Association, and the Association of American Medical Colleges. We also received a coalition letter signed by 63 organizations, including more than a dozen academic medical research centers, disease advocacy organizations, and others. These organizations have all written to express their support for the immense value that fetal tissue has brought and continues to bring to scientific research and medical advancement. Mr. Chairman, I ask unanimous consent that these letters be entered into the official hearing record.
Without objection, so ordered.
Thank you.
The chair recognizes the chairman of Subcommittee on Healthcare Benefits and Administrative Rules, the honorable gentleman from Ohio, Mr. Jordan.
I thank the chairman. I thank him for putting this hearing together today, and I'll be real brief. Look, we start from the premise, this is where the administration starts, that we want to be pro-life and pro-science. So I'm just looking forward to hearing from our witnesses about the life-affirming alternatives to fetal tissue research. My understanding is the technology is amazing, and there are ways to do this and get the results that we all hope to achieve. So I just look forward to hearing from our witnesses, and with that, I would yield back to the chairman.
I thank the gentleman. I want to go ahead and now recognize and introduce our witnesses. We have Dr. Tara Sander Lee. Dr. Sander Lee is an associate scholar at Charlotte Lozier, and she is a molecular and cell biologist with 20 years of academic and clinical experience. Her research and genetic testing has focused on childhood diseases, giving Dr. Sander Lee a unique perspective on the use of human tissue in medical research. She received her PhD in biochemistry from the Medical College of Wisconsin and completed her postdoctoral training at Harvard Medical School. Welcome, Dr. Sander Lee. Dr. David Prentice. Dr. David Prentice is the chair and scientific member of the advisory board for the Midwest Stem Cell Therapy Center in Kansas. He's an expert on stem cell research and bioethics. In addition, Dr. Prentice has written numerous articles and lectured extensively about the science and ethics of stem cell research and biotechnology. He was also selected by President George W. Bush as President's Council on Bioethics to write a comprehensive review of adult stem cell research for the Council's 2004 publication. Dr. Prentice received his PhD in biochemistry from the University of Kansas. Welcome, Dr. Prentice. Dr. Sally Temple is a board member and former president of the International Society of Stem Cell Research, and I welcome you as well. Thank you all for testifying. In order for us to allow time for questions and answers, if you would please limit your oral testimony to 5 minutes. And Dr. Sander Lee, you're recognized first.
To the distinguished chairs and honored members of the committee, thank you for the opportunity.
I have messed up. Hold on. If you'll all stand up, and this is not your first rodeo, it looks like it's mine, and it's not. So if you'll raise your right hand. Do you solemnly swear or affirm that the testimony you're about to give is the truth, the whole truth, and nothing but the truth, so help you God? All right. Let the record reflect that they all the witnesses answered in the affirmative. And now, Dr. Sander Lee will go ahead.
Thank you. To the distinguished chairs and honored members of the committee, thank you for the opportunity to testify before you today regarding alternatives for fetal tissue research. As the chairman mentioned, my name is Tara Sander Lee. I have extensive training and experience related to the use of human tissue in research, which can be found in my written testimony and CV. I stand before you today with a message of hope. We do not need fetal body parts from aborted fetuses to achieve future scientific and medical advancements. Several alternatives exist that are abundant and more successful, After over 100 years of research, no therapies have been discovered or developed that require aborted fetal tissue. History has shown us that we never needed fetal tissue to begin with. In the case of vaccines, cells derived from aborted fetal tissue have been used in the development process, but fetal tissues have never been the exclusive means necessary for these breakthroughs. What does this mean? This means that if we stop harvesting fresh tissues from aborted fetuses today, it will not stop one person from being treated or vaccinated, nor will it inhibit the development of new vaccines or therapies going forward. Case in point, development of the original polio vaccine was first produced in monkey kidney cells. Most polio vaccines currently use the cell line Vera, or monkey, in the manufacturing process. Measles, mumps, and rubella vaccines can all be produced in non-fetal cell lines. And none of the 75 vaccines available in the U.S. are produced using fresh fetal tissue. In addition, fetal tissue is not critical to the development of new vaccines to fight Zika and Ebola. Several ethical alternatives are available to researchers now for studying the most complex processes, such as brain development and neurodevelopmental disorders, immune response to pathogens such as HIV and Zika, and stroke. These models include, but are not limited to, fresh human tissues from adult and pediatric populations, donated and discarded biopsies and surgical specimens from both living and post-mortem individuals. Researchers have access to these ethical human tissue sources, which are available at several hospitals, academic institutions, and tissue banks across the nation with proper consent and IRB approval. For example, discarded cord blood from labor and delivery is stored in tissue repositories because it is a rich and plentiful source of stem cells for research, regenerative medicine, and transplantation. Stem cells from placenta, amniotic fluid, and even a mother's own bone marrow are even showing tremendous potential for in utero stem cell transplantation and treating the unborn before birth. In addition to stem cells, fully differentiated somatic cells from tissues such as skin can be reprogrammed to make induced pluripotent stem cells, or iPS cells, that possess an embryonic-like state. These pluripotent stem cells can then be further reprogrammed to generate organoids, also known as organs in a dish, which are 3D cellular structures replicating normal organ function in vitro for brain, liver, pancreas, intestine, stomach, lung, kidney, and eye. These organoids have proven successful in modeling complex mechanisms, including Zika virus and microencephaly, brain disorders, retinal development, infectious disease, cancer, and many others. Post-mortem donations are another useful solution. One option is cadaveric tissue, from which stem cells can be isolated up to several days after death. Finally, we do not need fresh fetal tissue from humanized mice because alternatives are available and being used now. Surplus human thymus tissue from newborn babies obtained during surgical procedures, as well as CD34 positive hematopoietic or blood stem cells from cord blood, adult bone marrow, and GCSF mobilized peripheral blood are all successful alternatives. In fact, human cord blood is perhaps the best scientific and non-controversial source for optimal human cell engraftment because it is abundant and more efficient and clinically relevant with respect to immune response. Other options include the ability of researchers to actually purchase humanized mice directly from commercial vendors such as Jackson Laboratories for immediate shipment. Such models include the human CD34 positive NSG humanized mouse developed using CD34 positive stem cells from cord blood, again without the use of human fetal tissue, as well as peripheral blood mononuclear cells for short-term experiments requiring mature T cells, including HIV infection. In conclusion, it is clearly evident that numerous alternatives exist and are available to researchers for studying some of the most complex diseases and disorders facing our nation today. I urge Congress and the Trump administration to stop using federal tax dollars to support aborted fetal tissue research and redirect funding to these alternatives. Thank you.
Thank you, Dr. Sandra Lee. Dr. Prentice.
Thank you, Mr. Chairman. I'm a cell and developmental biologist testifying on my own behalf and at the request of the chairman. Associations and positions noted are for identification purposes only. I've been a professor and researcher for approximately 40 years. Mr. Chairman, there's no scientific necessity for the continued taxpayer funding of fresh fetal tissue. Ample scientific alternatives exist, and modern alternatives have overtaken any need for fresh fetal tissue. Taxpayer funding, which is what this discussion is really about, should go to successful, patient-focused alternatives. One of those alternatives, adult stem cell transplants for patients. As of December 2012, one report found that over 1 million patients had been treated with adult stem cells, and that was actually an underestimate. A public face for such patients can be found at the educational website stemcellresearchfacts.org. Adult stem cells have been successfully applied for the treatment and recovery from various cancers, but there's still much to be done. One groundbreaking study has led to a first-of-its-kind clinical trial at the University of Kansas Medical Center with help from the Kansas Midwest Stem Cell Therapy Center to treat graft versus host disease, and there's initial success in this trial. Beyond cancer, adult stem cells are also showing therapeutic promise for other diseases and conditions where previously there had been no available treatment option. A couple of those examples: stroke and the damage from that traumatic brain injury. A groundbreaking study showed recovery of patients even years after the stroke event, and it's now led to a nationwide Phase 2B study for safety and efficacy. Again, one of those sites is in Kansas. An autoimmune disease that's showing significant strides in treatment using adult stem cells is multiple sclerosis, and actually putting patients into remission. One of those patients, Allison Carr, we just released a video documenting her treatment with adult stem cells. She was told by 2 doctors that she had 5 years before she would be disabled and on a feeding tube. After her adult stem cell transplant, Allison is now out riding horses with her kids, living a full and active life. Adult stem cells, including umbilical cord blood stem cells, have also been applied for conditions such as sickle cell anemia, not just for children now, but also being applied to adults. In one case recently, adult stem cells genetically corrected from the patient themselves have been used to treat a young boy in Germany suffering from a genetic condition and replacing all of his skin with healthy skin. Adult stem cells are also being used now for treatments while still in the womb, including for conditions such as thalassemia, a blood disorder. Mr. Chairman, we could go on and on about these examples, millions of which exist. I wanted to make a couple of other points, though, in the time I have remaining. One is the distinction between fresh fetal tissue and these historic fetal cell lines. Fresh fetal tissue is derived from ongoing abortion, and that's what the federal taxpayer funding debate is about right now. It's not about these historic cell lines. Those lines were derived in the 60s and 70s from an abortion. But the cells that have been kept in culture for decades now are not fetal tissue. They're cell lines, and that's not under debate. There are actually better ways to make vaccines now than through those historic cell lines. Some examples include the new Shingrix vaccine for shingles, which is made in a non-fetal type of cell and is actually better than the earlier shingles vaccine made in fetal tissue. Another example is the Ebola vaccine, which now been applied over 40,000 doses in this recent outbreak of Ebola in Africa successfully. The earlier study showed 100% protection of this non-fetal derived vaccine. The use of organoids, as my colleague has indicated, have also been used to determine the mechanism of action of Zika virus on the developing brains that results in that microcephaly we saw. Mr. Chairman, there are many other points we could make, but in summary, I want to say that, again, there is no scientific necessity for continued use of fetal tissue. It prevents no advantage to medical research. Instead, we have modern, advanced science that can be deployed more successfully and give better answers. Taxpayer funds should be redirected to the numerous modern productive scientific alternatives. Thank you.
Thank you, Dr. Prentice. Dr. Temple, you're recognized.
Thank you. Chairman Meadows and Jordan and Ranking Member Krishnamurthy, thank you for the opportunity to testify at this hearing on the value of fetal tissue research. I am Scientific Director of the Neural Stem Cell Institute and here today on behalf of the International Society for Stem Cell Research, where I served as President. I offer my perspective as representative of nearly 4,000 research colleagues around the world and as a scientist who has spent 32 years pursuing research to fight human disease. Fetal tissue is an essential resource for study and developing therapies for cancer, HIV, Zika, tuberculosis, and other devastating diseases. Fetal tissues have unique properties. The alternatives mentioned may be useful at times, but cannot fully replace fetal tissue. Zika. Fetal tissue is key for studying developmental conditions, including birth defects associated with Zika. Through studying donated fetal tissue, we're learning how Zika infects specific types of brain cells, producing malformations such as microcephaly that can severely impact quality and length of life. This research would not have been possible with other research models because we cannot mimic normal fetal brain tissue using current stem cell technology, including brain organoids. HIV/AIDS. HIV is a human pathogen that attacks the immune system. Animals that model the human immune system are essential for HIV research. The development of Truvada, an antiretroviral therapy that prevents the transmission of HIV, depended on fetal tissue. These animal models, mimicking the human system, are also crucial for ongoing drug discovery for dengue, influenza, Ebola, tuberculosis, and other infectious diseases. The alternatives mentioned to this fetal tissue model are not enough. The so-called neothigh mouse that uses the surplus thymus does not model the full and long-term development of human blood cells, including the immune cells that are relevant to viruses like Zika and HIV. Cancer. Fetal tissue research is crucial for understanding why and how many childhood cancers develop. Recent research compared the stages of fetal retinal development to samples from children's eye cancers to identify the genes responsible for tumor formation and growth, opening new possibilities for drug discovery. Similarly, fetal tissue is helping scientists target genetic mutations responsible for children's muscle cancers. CAR-T immunotherapy, which has revolutionized cancer therapy, was made possible after decades of fetal tissue research. The development of these products would not have been possible without testing them in mice that closely model the human immune system. Our ability to develop the next generation of immune therapies for other types of cancer depends on fetal tissue-derived mouse models. Alternatives to fetal tissue are not sufficient. All of them come with significant deficiencies. Tissues from miscarriages frequently have abnormalities. Fetal cell lines are typically cancer-like and often composed of a single cell type rather than the many cell types present in fetal tissue. iPS-derived organoids do not contain the full mixture of cells or fully mimic tissue structure. Adult stem cells cannot be used for every disorder, and it is dangerous to suggest this is the case. To close, I would like to reiterate that fetal tissue is critical for biomedical research to develop treatment for many human diseases. Alternatives cannot be proven to work unless they've been compared to fetal tissue. Without validation, patients are put at risk. Fetal tissue remains essential and it is simply wrong to suggest otherwise. Thank you. I look forward to taking questions.
Thank you, Dr. Temple. I'm sure that you'll get a few questions about your opening testimony, so I'm going to go to the gentleman from Georgia first, gentleman, Mr. Heiss.
Thank you, Mr. Chairman, and I appreciate each of you being here, and thanks for holding this hearing. Drs. Lee and Prentice, I really appreciate the strong manner in which you declared that aborted fetal tissue in general, is not necessary when we have the alternatives before us for scientific research, and that no one will suffer for lack of vaccines and other issues similarly if the fetal tissue research is not taking place. So let me just kind of run on that, because obviously the 800-pound gorilla in the room is we know that aborted fetal tissue is being used. We do know that abortion providers, for an example, Planned Parenthood or whatever, performs an abortion, then they sell the body parts to a middleman company who then sells it to research institutions or whatever, and oftentimes, I'm afraid, federal grants are used in this process. Is that the general method as far as you understand?
Yes, that's exactly the method by which people obtain fetal tissue.
Okay. Now, so we know this process is still going on. Are you at all familiar with the type of claims that are made by abortion providers on the informed consent forms that they give out in terms of the type of research that is done with fetal tissues and the diseases that supposedly it helps?
Yes, Congressman. In fact, I have a copy of a consent form from one of those facilities, and if I might read it into the record, it says, research using the blood from pregnant women and tissue that has been aborted has been used to treat and find a cure for such diseases as diabetes, Parkinson's disease, Alzheimer's disease, cancer, and AIDS.
I didn't know we had a cure for any of those.
It's my understanding we don't,
Okay.
Congressman.
Well, that would be my understanding as well, and yet, so is it fair to say that some of that information is at best misleading or inaccurate?
I'd say that's a fair statement. In fact, when this was presented to a group where I was at, where various ethicists and scientists were read that statement, all of them, to a person, said that particular statement would not have passed their institutional review board as being a fair and full consent.
So what do you think that tells us about the business of fetal tissue procurement?
My read of that, Congressman, would be that it's misleading, certainly, in terms of making people think that there have been great strides made with fetal tissue. And again, my contention would be that it has not been very successful medically, and that there are many other alternatives that are better science.
Okay, so let's get to a little more specifics that perhaps we don't want to say. When a mother agrees to donate an aborted baby, how does the abortion provider then harvest the fetal tissue and organs? Dr. Lee?
So the abortion provider so the abortion provider will provide the actual fetus to a third-party vendor, such as ABR or Stem Cell Express, and then they will harvest the tissues and isolate the cells that they want, and then that's what they can sell.
Okay, and they do sell?
They do sell them.
Supposedly, these parts are not to be sold for valuable consideration. In other words, they're not to be a profitable sale. And yet we all know that's not happening. In fact, a year ago this month, 2 companies in California had to pay almost $8 million for selling a baby liver, $350, the thymus, $500. We can go on and on and on with this. I think a lot of times, though, profit is made in the transporting of these body parts to try to get around the law. It seems like there's a real weakness in our law protecting the valuable consideration. Would you agree with me in that regard?
I believe that there are some serious concerns with how they are charging some of these fees.
Dr. Prentice, would you agree?
Yes, I'd agree.
Okay. Well, using the alternatives that you talk about, that both of you have mentioned that have been so successful to the extent that you believe that the fetal tissue is not even needed for scientific research, I'd like to get into, what's the cost comparison? Let's go this route. The cost comparison between the alternative use versus the fetal tissue use of research.
I can give you one example just in regards to if you want to use humanized mice to test immune response. So the current, one of the most popular methods, as we know, is to obtain the fetal liver and the fetal tissue, and the fetal liver and the fetal thymus to generate these human mice from the fetus. But it's been shown in a recent manuscript that if you obtain neonatal thymus, that this is actually more efficient and more abundant. So that in the end is going to mean that it's going to cost less. So for example, the authors state that it's more abundant because more than 1,000 fragments can be obtained from one donor. This compares to fetal liver, which you can only obtain 20 fragments per aborted fetus. So that means that they can engraft 50 times more mice per donor with neonatal tissue versus fetal tissue. So just in that one example, it's going to be a cost.
Dr. Prentice, do you have anything you'd like to add to that?
Not at this time.
Okay. Listen, I appreciate there's a lot more directions we can go with all of this, but I appreciate the fact that you both stated emphatically that tax dollars do not need to go further towards the fetal tissue research, and I am deeply appreciating your commitment in that regard. It seems to me that there's a lot of talk these days about a wall being built. I think emphatically there should be a wall built between the taxpayer dollars and the use of those dollars going toward aborted baby parts and the research thereof, and I appreciate the fact that you are giving testimony to Congress in that regard as well. And Mr. Chairman, I yield back.
I thank the gentleman. The chair recognizes the ranking member, Mr. Krishnamurthy, for his line of questioning.
Thank you, Mr. Chair. You did well. Thank you. I'm troubled by recent actions the Trump administration has taken to undermine fetal tissue research, because these efforts, I fear, hamper our ability to research the cures or vaccines or therapies for afflictions that, basically tear apart the lives of millions of people around the world. I wanted to ask Dr. Temple a couple questions. According to press reports, the Trump administration has quietly ordered scientists employed by the National Institutes of Health to stop acquiring new fetal tissue for their research. Dr. Temple, in your opinion, how could the administration's order to stop acquiring fetal tissue affect ongoing biomedical research at the NIH?
So I think this is a very serious concern. The NIH is viewed around the world as being preeminent research and leading research organization. The work that they do benefits the entire community. And if the work on fetal research and fetal tissue is stopped, what that means is that we are halting very important research into cancer, into HIV, into children's tumors, and the cost of this is going to be huge.
Do you have a sense of, have you spoken with researchers about this particular topic, and what are they saying with regard to potentially using humanized mice or organoids or adult stem cells in place of fetal tissue for those particular research projects?
Yes, so I have spoken to many of my colleagues, and we all agree that these alternatives are not sufficient. This is cutting-edge, very important progressive research that uses the humanized models. For example, we need to have a model of the human immune system to develop new CAR T therapies and to develop new HIV therapies, eventually leading to a cure for HIV/AIDS. These would not be possible without this resource.
So just to put it in layperson's terms, are you saying that the fetal tissue possesses certain qualities which these other sources do not?
Absolutely, yes. It has unique properties that these other alternatives do not adequately.
And can you just lay out what those properties are so that we understand?
Yes. So, for example, in creating the humanized mouse model, it's essential that you have human immune cells and they are repopulated for a long period of time so that we are able to study how the immune system interacts, say, with cancers or interacts with infections that target the immune system. The alternatives that have been talked about today do not do that. For example, the neothigh mouse does not replicate all of the cell types in the immune system that are required to model and understand these diseases.
Okay, but how about adult stem cells?
Adult stem cells are very restricted. They can do just a few things. They may be applied, for example, for skin, for blood, but adult stem cells do not efficiently make other organs. They also do not necessarily tell us about fetal development. For that, we must turn to fetal tissue to learn about that.
Okay. What, in your opinion, is going to happen? You know, it's been 2 years now, or it's been some time now since this order was put in place. You know, what does a lost year of research mean in the fields that the NIH is doing research?
So I have, again, spoken to many of my colleagues, and the word that they have used over again is devastating, that this will essentially cripple many of the important lines of investigation and therapy development ongoing.
Thank you.
I thank the gentleman. So, Dr. Temple, let me make sure I understand. So you're saying fetal stem cells have been more successful than adult stem cells in terms of therapy? Is that your sworn testimony here today?
What I'm saying is that you have to look at the entire research enterprise. Adult stem cells have been used for decades.
How many successful fetal stem cell successes would you say we have today?
I can talk about Truvada, which is important for preventing HIV passage. I can talk about the development of retinoblastoma drugs, and also retinitis.
And those are successes, is what you're saying?
These are in progress.
I thought some of those were still in clinical trials.
They are in progress, but they are successful.
Well, so how do you know whether, listen, I know clinical trials. If they haven't finished the clinical trial, how can you say they're successful, Dr. Temple?
Because
it's sworn testimony. I want to make sure that you understand that.
Oh, successful, I mean successful in terms of progress.
Okay, all right. I'll recognize the gentleman from Ohio, Mr. Jordan.
I thank you, Mr. Chairman. Dr. Lee, 20 years you've been in this field, looking at your resume, Medical College of Wisconsin, which I appreciate as a UW graduate myself, PhD in biochemistry, trained at Harvard Medical School, Boston Children's Hospital. Pretty impressive. You know what you're talking about, don't you?
I believe I do.
Yeah, I do too. And you said in your testimony, we do not need fetal body parts from aborted babies to achieve future scientific and medical advancements. You stand by what you said in your testimony, right?
I do.
And you also said after 100 years of research, directly to where the chairman was with Dr. Temple, you said after over 100 years of research, no therapies have been discovered or developed that require aborted fetal tissue. Is that accurate?
That is accurate.
And yet, taxpayers are given money paying for this fetal tissue every single year. Is that true?
That's true.
let's start with Planned Parenthood. You know how much money Planned Parenthood gets from taxpayers in a year's time? Do you happen to know?
They get about $500 million.
It is about $500 million. Several million directly, and then some of that, of course, is Medicaid funding as well. How much is the government paying for fetal tissue research?
Approximately $100 million.
$100 million a year we're paying for fetal tissue research, and how much of that is involved with actual experimentation on the fetal tissue?
$20 million. Approximately $20 million if you look at NIH contracts.
About $20 million, okay, and that's NIH. That's not HHI. That's NIH.
That's correct.
Okay. Is some of that $100 million, some of that $20 million, is some of it actually used to purchase the fetal tissue?
That money is going to be used to purchase the fetal tissue.
And I think with Mr. Congressman Heiss earlier, you said it's not a direct purchase, though, right?
It's
In other
not.
words, so let me just, I just want to understand it and make it as simple terms as we can. Planned Parenthood is taking the life of unborn children. Planned Parenthood then, or other abortion providers, sell the tissue to some company. And then the government then buys it from that company. Is that accurate?
That's accurate.
And who are some of those companies? I think you mentioned some names in your...
A couple of them are ABR and Stem Cell Express.
ABR and Stem Cell Express. Any chance those companies were involved? There's some somewhat now famous videos that Planned Parenthood had. They didn't have, but they were about them a few years ago. Stem Cell Express and ABR, were they involved in any of those videos of Planned Parenthood a few years ago that somewhat now well known?
They are. They were absolutely, they were shown, their question as to whether there was profit actually being made, and because there were actual records that they were only paying approximately $60 to Planned Parenthood or other abortion providers to get the fetus, and then they would turn it around and then they would sell it to investigators and make much more money than that.
So I just want to be clear again. So those videos that we saw that we're all repulsed by, that were disgusting, all kinds of this cavalier attitude that people at Planned Parenthood were talking about, when they were doing that, talking in those transactions, it was with those companies you just said, Stem Express and ABR?
Yes.
Okay. So again, here's how it works. Taxpayer dollars, our constituents' tax money is going to Planned Parenthood. Planned Parenthood then provides abortion services. When they then sell fetal tissue to Stem Express, ABR, and companies like those who were involved in those videos that we saw, and then the government then takes taxpayer dollars and buys the fetal tissue from those companies.
That's correct.
To do research that has not in 100 years provided anything, what was your words? In over 100 years, no therapies have been discovered or developed that require aborted fetal tissue.
That's correct.
And all we're asking is, let's go with the alternatives, which you have said are actually better.
That's correct.
Well, that's as simple as it gets, right?
Yes, it does.
I mean, you can't make it any plainer than that. Taxpayers are paying on the front end for something that many of the folks I get the privilege of representing just think is absolutely wrong, giving money to Planned Parenthood, which is taking the life of unborn kids, and then they're paying on the back end for the very tissue that was used in a practice that they don't approve of to get results that can be achieved with alternatives. And according to your 20 years in this field, after 100 years of this kind of research, no discoveries, no therapies have been discovered or developed that require the actual use of fetal tissue.
That's correct.
Well, this is pretty simple.
It is.
I mean, shoot, Chairman, we can stop. We got it right here. I'm sure there are other people who have questions, but that is as plain as it gets,
Yeah.
right?
That's right.
Taxpayers, something they don't like, both on the front end and back end, are paying for it, and we do not need the fetal body parts from aborted babies to achieve scientific and medical achievements because we have alternatives that can do it.
That's right.
I'm glad you came, Dr. Lee, and more importantly, I appreciate the work you've done now for over 20 years in this area. Thank you.
Thank you.
All right, they have called votes, and so we're going to recess subject to the call of the chair, but for planning purposes, we will not be long. So I would think that we would be back in and reconvening right after votes, no later than 11 o'clock.
sound check for Mr. Fowler. She quotes the letter, but this is just advice.
Let me check.
Yeah, my boss wants to enter to the record. If you have a copy.
Yeah, we're going to wait about two more minutes to see if Mr. Krishnamurthy is headed back, and if not, then we'll gavel in, just so you, two.
Thank you.
And since Mr. Jordan is ready for us to get we'll start on your side. You quote it, I'll decide. This whole second page doesn't matter. Neotype tissue is non-fetal tissue.
Okay, yeah.
The House, the Subcommittee on Government Operations and Health and Benefits and Regulations will come back to order. I thank you all for your patience. And the chair notices the presence of a few members that are not on the subcommittees and yet want to be part of this. And so I ask unanimous consent that they can be considered as part of this committee for questioning. And with that, the chair recognizes the gentlewoman from the District of Columbia, Ms. Eleanor Holmes Norton.
Thank you very much, Mr. Chairman. And I've been watching these proceedings from my office. And I did notice that my colleagues were once again raising the long-settled issue of selling fetal tissue from abortions for profit. Now, the select command, I just want to therefore put into the record the select panel on infant lives, which spent $1.5 million looking into this very question. They found no credible evidence of wrongdoing. So to have this raised again and again, no matter what the evidence, is just too bad. They interviewed the organizations that provide fetal tissue to researchers, recognizing that federal law does allow for the reimbursement of reasonable expenses for tissue samples. I'd like to quote, there was a Democratic report and a Republican report. I'd like to just report from the Democratic report. These companies consistently explained that costs related to fetal tissue procurement outweigh revenue that they receive for this service. I don't know what more evidence is needed. I'd like to ask a question, I believe it is Dr. Temple, on what special role fetal tissue still may play. We've heard about the critical role in Zika. Dr. Temple, can you give us a few other examples? I'm particularly interested in childhood diseases that benefited from the use of fetal tissue research.
Absolutely. And I think I'd like to start by also referring back to Mr. Meadows' comment on how what medicines and valuable information has been gained from fetal tissue research. It's important to understand that the way that the research process works is that we learn and study about the biological material, and then from there we develop medicines. And that process is long, it's difficult, but it is already demonstrated valuable products. For example, Truvada, which is to prevent the transmission of HIV in the use of PrEP, that depended on the use of the fetal tissue humanized mice. And it is incorrect to say that advances that have gone on did not rely on fetal tissue. That is not the case. In terms of childhood diseases, there are diseases of the retina, for example, in which understanding the biology of the developing retina has enabled us to learn about how those childhood tumors develop, and blinding diseases and disorders such as retinoblastoma. So also cancers of the muscle that occur in childhood. These cancers arise during fetal development, so we must study fetal development in order to understand that tumor process.
That's important, that fetal tissue, excuse me, I have a cold. Why fetal tissue is used to study childhood diseases, particularly those in utero.
Absolutely.
Yes,
may
please.
I add? But not only that, the need to develop models that have the most complete human immune system is essential for studying viruses such as Zika, such as HIV, in which the cells are infected, the immune cells are targeted, and therefore, because this is a human pathogen, we need the human cells in order to do that. And currently, the best models are the ones that use fetal tissue, the most complete models. And the other models that have been spoken about today do not fulfill that need, and therefore they cannot be used as an alternative.
In his written testimony, Dr. Prentice alluded to a number of instances, excuse me, in which adult stem cells are being used to treat diseases in utero. Dr. Temple, I'd ask your view on that, whether fetal tissue can be replaced by adult stem cells or, excuse me, or other alternatives in the examples that you provided.
So no, they cannot. I actually work on adult stem cells. I know a lot about them. There are different types, but they cannot be used to develop therapies for everything. They have value, but it is limited, and they do not fully substitute for fetal tissue in either the study of biological processes and the development of specific new therapies that are ongoing and cutting edge right now.
Dr. Sandy Lee, and here I'm quoting from her testimony, if we continue to invest in fetal tissue research, we will delay future progress and advances using better and more ethical science. Do you agree with that? Is there a delay involved here? If you agree with it or not agree with it, I wish you would explain your response.
So I do not agree with it. We know, for example, that the models that are developed with the thymus tissue use very difficult to construct, and they do not model the immune system that I have described is absolutely essential. The cell lines that are used to make vaccines are valuable, but in order to make the original prototype vaccine, that requires these humanized models in many cases. We're not talking about the vaccine manufacturing, in which fetal cell lines or other lines may be useful, but the original research upon which that was based depended on fetal tissue research, and new vaccines similarly do so.
Thank you, Dr. Hempel.
The chair recognizes the gentleman from Wisconsin, Mr. Grothman.
Thank you very much. First question is for Dr. Prentice. You don't use fetal tissue at the Midwest Stem Cell Therapy Center, correct?
No, Congressman.
Has this prohibited you from doing cutting-edge research?
None of the research at the Midwest Stem Cell Therapy Center uses aborted fetal tissue, but they have made great strides in spite of that.
Yep. Can you tell me examples?
Probably because of that.
That's interesting that you say because of it. And there are other centers like yours around the country. Can you explain why, first of all, some of the progress that you've been making, and secondly, why you feel you're being able to make progress at a greater rate than some of the centers who feel they have to use fetal tissue?
Well, the origin of that not using the aborted fetal tissue, frankly, was from the legislature that initially set up the Midwest Stem Cell Therapy Center. And part of the reason was to focus on the more successful adult stem cells and the potential treatments to get this into the clinic faster for the patients. So I mentioned one of the clinical trials. It's a homegrown clinical trial, first of its kind, to treat graft versus host disease using an adult stem cell source. And that clinical trial is still ongoing, so this is very early, but the initial results seem very successful. In association with the Bone Marrow Transplant Center, they have been treating a number of different cancers as well as anemias, including sickle cell anemia. They have also engaged in this stroke study to treat patients who have experienced stroke even years in the past.
Overall, countrywide, has there been more progress with adult stem cells or fetal tissue research?
There's much more.
Not even close, right?
I'm sorry?
Not even close.
Not even close.
Why do you think when it's not even close, there's some people out there who insist? Is there something psychological going on, this apparent love affair with fetal tissue? Why psychologically would people prefer to use that than the more effective adult stem cells?
Well, I think in some cases, as a scientist, we're trained in a certain way, and we may be reluctant to switch to something new, something that, even if we know that it's better and potentially more productive. But, Congressman, I'd like to make a point about that when we do step out, great things can happen. And if I could give you just one brief example, if we look in the stem cell field at the production of these induced pluripotent stem cells, the Japanese scientist Dr. Yamanaka came up with a few years ago. One of his reasons, frankly, was he'd looked through the microscope at an embryo in a colleague's lab and said, you know, we can't see much difference between this and my daughters. We can't keep destroying embryos. There must be a better way to get the same kind of cell. And he came up with these iPS cells, which mimic pretty much everything that an embryonic stem cell can do, and won the 2012 Nobel Prize for it.
Okay. I sometimes wonder when it comes to politicians who are always pushing the fetal tissue, if it becomes because they've been so invested in abortions and they want abortions to be good that they just want to believe that fetal tissue research is superior, and they have this psychological problem. But I don't want to let you alone, Dr. Sanderly. Can you tell us a little bit about reprogramming adult stem cells into pluripotent cells?
Yes, so you can generate what's called induced pluripotent stem cells from somatic cells. So for example, you could take a skin biopsy from an adult human, and you could isolate the skin cells, and then you could reprogram them. You could give them specific factors that would allow them to differentiate and basically revert back to an embryonic-like state. And then those cells, those pluripotent cells, can then be further reprogrammed into what are called organoids, which are basically simply organs in a dish that can be studied and have comparable, similar cell components to what we see in the fetus.
Can you give me examples of some sort of treatments that are evolving out of this?
So, for example, probably some of the best examples are when we look at brain development. So there are multiple publications in the literature which show that you can directly take skin fibroblasts from adults. You can actually even purchase these skin fibroblasts from biorepositories such as Coriell. You can make these iPS-derived forebrain organoids, which you can use to study Miller-Decker syndrome. There's actually key publications that have shown that you can take these cells and you can actually make cerebral organoids, and you can actually look specifically at astrocytes, and they have shown these astrocytes actually have functional pathways similar to the adult human cortex. We have other examples such as Luau and L from 2016 in which they've generated these organoids to recapitulate many of the features of the mid-fetal human brain. There's other examples for example, Monser et al. From 2017, in which they've taken these brain organoids and they've actually been able to graft them into a mouse and actually look at functional neuronal networks and vascularization, such as blood vessels. So these are all critical studies. And another example, Levine was able to actually take some of these. Actually, Zhang and Al were able to actually take these and actually research human developmental brain by recapitulating the medial ganglion organoids and cortical organoids. So there are multiple examples. I could keep talking just about how, and this is just brain development. This is even talking about retinal development, kidney development, and these are so key studies that can be done looking at multiple areas of development in the fetus.
Thank you, and
Mr.
I'd like to
Mathers.
thank the chairman for letting me talk.
Yeah, I thank the gentleman. The chair recognizes the ranking member of the Government Operations Subcommittee, Mr. Conley.
I thank the chair, and I know you're next. Actually, I'm simply going to insert in the record my opening statement. I won't read it, other than to point out, as I know Dr. Temple was going to try to point out, the idea that stem cell research can do it all is not true. And we also know that, frankly, fetal tissues research can save lives and can be pioneering in dealing with lots of diseases and conditions that are life-saving, potentially. And so, if we want to talk about being pro-life, there's another side to that, and that is trying to save lives, both prospectively and those that are already here, from horrible conditions and diseases. And one of the examples in my opening statement has to do with the efficacy of Truvada, for example, a life-saving drug that with which human tissues, fetal tissue, actually made a huge difference. So in any event, I would ask to enter my opening statement into the record, and I thank the chairman for his consideration.
I thank the gentleman. The chair recognizes Ms. Watson-Coleman for her line of questioning.
Thank you, Mr. Chairman, and I want to thank the witnesses for being here today. I want to start by saying how disappointed I am that I'm even sitting here today on this issue. Last Congress, I sat on the select investigative panel that was mentioned by my former colleague that wasted $1.5 million in taxpayer money on a witch hunt that incessantly attacked women's health and tried to intimidate doctors out of giving care. This hearing is yet another callous attempt to obstruct women's access to proper care and to impede life-saving research. I'd like to focus my questions on how the use of fetal tissue is contributing to the fight against HIV/AIDS. Earlier this month, as we commemorated National AIDS Day, we mourned the loss of family and friends and community members who are facing and who have passed at the hands of this terrible disease. According to the Centers for Disease Control and Prevention, in 2016, just shy of 40,000 people received an HIV diagnosis in the United States. Despite progress made in recent years, HIV/AIDS remains one of our nation's most pressing public health issues. one that disproportionately impacts the LGBT community and the communities of color. Dr. Temple, in your opening statement, you talked about the importance of fetal tissue research in developing the HIV/AIDS treatment, including the prophylactic treatment Truvada. In their opening statements, Dr. Prentice and Dr. Sandra Lee assert that the other types of tissue, for example, what is known as neo-thigh tissue, can in fact replace fetal tissue in efforts to humanize mice. Could you please explain or just restate whether or not these claims are correct or not?
Those claims are not correct.
In fact, the committee received a letter just this morning, Doctor, from Dr. Michael Brown, the author of a paper on humanized mice that Dr. Prentice and Dr. Sandra Lee pointed to in their statements. And he wrote, as I quote, "It is premature to take generalizable conclusions about the neo-thigh replacing fetal tissue, BLT type, models in all humanized mouse research applications. Further, in no way does our paper, his paper, support the claim that fetal tissue research as a whole is no longer needed. To that extent, I'd like to unanimous consent to enter this letter dated December the 13th into the official record."
Without objection.
Thank you, sir. Dr. Temple, does it surprise you that the author of the paper cited in Dr. Prentice and Dr. Lee Sanders' testimony wrote about the importance of fetal tissue in humanized mice?
Does it surprise me? Could you say it again, please?
Does it surprise you that the author of the paper cited in Dr. Prentice and Dr. Sandra Lee's testimonies wrote about the importance of fetal tissue humanized mice?
No, because this is the consensus of our field, that it is essential and cannot be substituted by such models, new models, model that has not yet been tested for HIV research.
Thank you, Dr.
For
Temple.
example.
This past September, the Trump administration ordered scientists employed by the National Institutes of Health to stop obtaining fetal tissue. This order has reportedly disrupted the HIV research using humanized mice at the Montana-based Rocky Mountains laboratories. According to scientists involved in this research, even if they were given the green light to resume acquiring necessary fetal tissue, it would take them a year to get their research back on track. How does this limiting access to fetal tissue undermine the work of the scientific community? Dr. Temple.
Yes. So any delay or restriction on this research will have devastating consequences on current programs that are using the fetal tissue to develop new cancer therapies, to develop new vaccines. And I want to explain that in a comment to an earlier point, that scientists do not do this because they are wedded to an old or outdated model. The models are the only ones that we have currently that will do what we need in order to research into the diseases and into the therapy development. And I also am an expert in brain organoids, and they do not recapitulate the fetal tissue. They are not complete. Unfortunately, I do this every day, and so I know this very well. They are not a full model of brain development and cannot be used as such.
Thank you. And Mr. Chairman, I simply want to say that as a grandmother of an autistic child, as a diabetic myself, as a recent cancer patient myself, I am personally engaged and committed to all kinds of research that will help us to overcome these devastated diseases or to help us to live with them because new therapies are developed. And I believe that the science is where we need to follow. And that while I know that this administration doesn't like the idea of science, whether it's climate change or fetal tissue, the truth of the matter is that science is important here. and I support the work that is being done here and will continue to do so. With that, thank you, Mr. Chairman. I yield back, and I thank you for your indulgence of 1.14 minutes.
I thank the gentlewoman, and I wish her well in her battle against I wish you well, and we'll be praying for you in your battle against this dreaded disease. All right, all right. The chair recognizes the president of the presence of Ms. Chu and Ms. Fox, and we ask unanimous consent that they be included as members of the subcommittee. And Ms. Fox is actually returning, so she is up next. So I recognize the gentlewoman from North Carolina, Ms. Fox. Dr. Fox, excuse me.
Thank you, Mr. Chairman. I appreciate it. And I, too, wish my colleague well. I want to thank our witnesses for being here today. This is a very, very important issue, and I think it is something that we need to constantly be talking about and thinking about, because it is at the heart of the life issue, and that's very, very important to me. I have some questions. I hope they can be answered fairly succinctly. Dr. Sanderly and Dr. Prentice, some questions for you. Did the Obama administration make any attempt to develop moral alternatives to fetal tissue research? Dr. Sanderly?
Not to my knowledge.
Yeah, not to my knowledge either.
Okay, thank you. Do you think the Trump administration understands the inherent ethical issues related to testing new medical treatments using human fetal tissue from elective abortions?
Do I think that they support?
Do they understand the inherent ethical issues?
Yes, they do, absolutely.
Dr. Prentice?
Yes, I do.
Thank you very much. What role should private hospitals, medical communities, and other private sector entities play in ensuring researchers have timely access to life-affirming alternatives? And what role should the federal government play in making that possible? Dr. Prentice?
Congresswoman, one aspect would be forming some sort of donation network for the neonatal tissues or the adult tissues or the adult stem cells that can be used for these very effective and growing number of treatments.
Thank you. Dr. Sanderly?
I'd just like to add that not only has neonatal thymus tissue, which has been shown to withstand cryopreservation and banking, that could be one option to make it easily accessible for people to generate these humanized mice. But in addition, there are several examples in which cord blood alone can be used to generate these mice. So even if you wanted not look at the brown paper, there are many examples where cord blood, and which absolutely can be withstand cryopreservation. I mean, all you have to do is look at Be the Match Foundation, which stores this for clinical purposes. So cord blood.
May I interject, because it is not the case.
I'd like to finish talking, please. But cord blood can actually be injected directly into these mice and used to study HIV infection. So having, like as my colleague said, having some type of bank where we can have easy access to such tissues like this, surgical tissues, cord blood, easy access to these in a timely manner would have a tremendous impact on the research community in studying some of these horrible diseases such as HIV.
Right. Well, as a follow-up to what you just said, do you have an example of researchers using surgical tissue?
Well, one of the examples is what we've been talking about, using the neothigh mice. So actually obtaining surplus neonatal thymus tissue for children that go in for these surgeries required for heart surgeries, in order so that they can have easy access to the heart and the vessels. They have to remove the thymus, and so it's a routine part of that procedure. So that's just one example. I can give you another example in my own research. I was studying congenital heart disease, and yes, I could have obtained human fetal tissue to look at it, but instead we actually obtained discarded human valve tissue from children going in for routine surgical procedures that could use directly into our laboratory.
Thank you. And Dr. Prentice, what makes surgical tissue a viable alternative to fetal tissue? Dr. Sanderly has given one example. Do you have another example?
For the particular example we're talking about, it's quite abundant, actually. The way this tissue comes about or is made available through these surgeries are little kids soon after birth who have some cardiac problem. The cardiac surgeons routinely remove part or all of the thymus, just to expose the surgical field. And so one estimate I saw was something like 40,000 of these surgeries done in the U.S. every year. So there's quite a bit of this tissue available. Some of it's already been used to treat other conditions. It's a matter of just having access to the tissue.
Thank you very much. Again, this is an extremely important issue, and I'm so glad the committee is holding this hearing today to bring us up to date on the wonderful research and findings that are occurring. Thank you, Mr. Chairman.
Thank you, Dr. Fox. The chair recognizes a gentlewoman from California, Ms. Chu.
Thank you, Mr. Chair. In light of my Republican colleagues' mischaracterization of scientific efforts involving fetal tissue, I'd like to take a moment to provide some context about the history of fetal tissue research in this country. The federal government has actually funded fetal tissue research since the 1950s. So we're talking about nearly 70 years in which this has been funded. In fact, it was back in 1988 that President Ronald Reagan established the Human Fetal Tissue Transplantation Panel to evaluate the science behind fetal tissue research. This panel, which was chaired by a Republican, ultimately concluded that, and I quote, it was acceptable public policy, that is acceptable public policy to support transplant research with fetal tissue, unquote. Following this panel, Congress passed the NIH Health Revitalization Act in 1993. The law established rules for how fetal tissue research is conducted in the U.S. It passed with overwhelming support from both Democrats and Republicans. Fetal tissue research is also governed by extensive regulatory frameworks that govern the acquisition and the use of this tissue. This has been investigated by multiple committees, including the Select Investigative Panel, at the cost of millions of taxpayer dollars, and no wrongdoing has been found. Efforts by this administration and my colleagues on the other side of the aisle to undermine the integrity of the existing framework around fetal tissue procurement are motivated by extreme ideology. That's all it is. Perhaps even worse, they do great harm, in fact, extreme harm to science. Fetal tissue research has been recognized for the potential that it has to help millions of people. We must not forget that this critical research has a long history of support from both sides of the aisle. So, Dr. Temple, could you please explain some of the groundbreaking medical advancements prompted by fetal tissue research?
Absolutely. And before I do that, I just want to really comment that your comments were exactly right, and that these fetal tissue research has been enormously valuable. For example, it has laid the groundwork for therapies ongoing now in Parkinson's disease, research into Alzheimer's disease, HIV, AIDS, CAR T therapies. You know, last year, the third CAR T therapy was approved. This was very important news. This is a way now to treat people with lymphomas. I spoke to pioneers of that technology who explained that the fetal tissue mice that model the human immune system is absolutely critical for the development of new CAR T therapies against other forms of cancer. This view that you expressed that I agree with is really the consensus view in the scientific community. I have not talked to people who disagree with the value of fetal tissue research. I know we heard it from two individuals today, but this is not the consensus view. that the value of fetal tissue research is very well recognized, and we believe it can be done, as it has been for decades, in an ethical framework.
Thank you. Thank you, and I yield back.
I thank the gentlewoman, excuse me. The chair's going to recognize the gentleman from Texas, Mr. Olson. Ms. Temple, I want to make sure that I understood you correctly. Are you saying the only two people that you've ever talked to that believe that there is a problem with using fetal tissue are the two people seated to your right?
Today, that is the
You've
case.
never talked to anybody else
very,
that's had that problem?
With the fetal, to misrepresent the need for fetal tissue. No, in the scientific community.
I understand it's consensus. I got that. But you said you've never talked to anybody that has ever taken issue with that. And I just find that statement just extreme, to put it in the words of Ms. Chu.
But I should clarify that this is a minor view, perhaps amongst the scientific
But you have talked
community.
to other people other than the two people seated to your right that would share their view. Have you talked to anybody other than the two people seated to your right that share their view? Yes or no?
The majority of scientists.
That's not the question. You're answering a great question I didn't ask. Have you spoken to anyone other than these two people that would agree with them?
I do not recall doing so.
Okay, that's good to know. I'll recognize the gentleman from Texas, Mr. Olson.
Well, before I ask my questions, I want to thank the chairman, the ranking member, all the members of the committee, the staff, for giving me the honor to participate in this hearing involving issues with fetal tissue research. This issue is very important back home in Texas 22. Dr. Prentice, I read all 12 pages of written testimony last night. And while it can't compare to the Bible, I was saying amen over and over and over. Page 2, I quote, adult stem cells are also showing therapeutic promise for other diseases and conditions where there has previously been no available treatment option, end quote. Amen. Page 1, you offer, quote, a public face for such patients. Have I got the face for you? This is Sarah Hughes. She lives in Katy, Texas. She is my guest at Donald Trump's first speech before Congress. When she was 11 months old, she was diagnosed with systemic juvenile idiopathic arthritis. As you all know, that is a death sentence for this young lady. Her disease was treated with base-like cancer, drugs. The drugs made it even worse. She withered away. She did eat solid food for over 10 years because the body reject the food. God put her in touch with a young man who had an experience with Celtech's adult stem cells in Houston, Texas. What they did with Sarah is they take her own stem cells from her body's fat and spin it up and make a whole lot more cells and inject them back in Sarah's body. This was how she looked for her stem cell treatment. Her mom, Fiona, told me this picture was taken. She was probably 2 months or less from being called home by our Lord. Sarah had two problems with stem cells. First of all, despite the fact that these are her own cells, our FDA says this is a drug. It can't be rejected, but somehow it's a drug. So while we can harvest the stem cells in Houston, to inject her body, she has to go to Cancun, Mexico. The first injection she had, she said, I felt a change. And guess what happened within 2 years? I was at that young lady's wedding. She never, ever thought that would happen except for her adult stem cell therapy. So, Mr. Prentice, Texans love to brag. I see you've got some cowboy boots on, got some text in you. Could you brag other stories about Sarah? Is this isolated or is this commonplace, going more and more commonplace every day in America? Adult stem cells saving lives all across the spectrum with all these different diseases.
Well, Congressman, you've got a face there. I talked about a face. There are probably getting close to 2 million of those types of faces treated with adult stem cells now around the globe. And, you know, the story you told, Allison Carr's story, treating with her own adult stem cells for multiple sclerosis. Sonia Kuntz successfully treated for her stroke with adult stem cells. The list goes on and on and on. And we could, frankly, use up the committee's time, Mr. Chairman, to go through all of the various things. But the point is still that adult stem cells are the gold standard when it comes to stem cells for patients. And the only one showing such great success over and over and over, whether we're talking about Texas, Kansas, or anywhere around the globe.
So, Mr. Chairman, may I interject, because this really is a very important point. Adult stem cell therapy that has been provided by these clinics that offer unproven therapies and based on anecdotes have caused tremendous harm. For example, several women in Florida were blinded by this treatment. We have to be extremely cautious.
Dr. Temple, so I appreciate that.
Thank you.
And
This is
your
for patient
commentary
welfare.
but your commentary on Dr. Prentice's, I didn't allow him to make commentary on yours. And so if we're going to get a colloquy going here, we're just not going to do that, okay? And so because we also have proven evidence where we actually put in fetal tissue from a standpoint of some brain research that didn't turn out real well, as you're well aware of, right?
This is the case.
Yes.
Yes.
So,
yes.
so,
Again,
so we
that
we
was
need,
an
we need good science.
unproven
We need
therapy.
good science
Yeah.
that works through that. And so, since I jumped on, I'll give you one last question, Mr. Olson, then we've got to yield back.
A different topic here, but it's the entire panel. I know the entire world was horrified. We found out that in China, a doctor had modified embryos genetically and had babies born alive. Now, he has disappeared. That was condemned by China, every healthcare professional, ethical people across the world. But sadly, my alma mater, Rice University, had a role in that. He trained there, the Chinese doctor did, before he came back to China and did his evil deeds. So my question is, how can we get in front of this and make sure this never, ever happens again? Dr. Sandra Lee.
How can we get in front of this to make sure that something as unethical as that happens again? Well, when we have, as far, do you want me to speak specifically about the?
Sure.
Or do you want me to talk just in general about, I mean, my initial response is that when we are faced with other things in the country, such as fetal tissue that is non-controversial, that has a controversial issue, that's where we stop it. And so, but as far as this issue, I think my colleague actually has a lot of experience with this, and I'll pass that to him.
Okay, we're way over time, so very brief, please.
Congressman, one of the things I think...
Everything's bigger in Texas, and so is his time clock, so go ahead.
Yes, Mr. Chairman. Congressman, I think very quickly, one thing that we should probably look at is a moratorium on these sorts of experiments with embryos.
Great idea. Dr. Temple, 10 seconds, ma'am. 1,000.
Dr. Temple, I think he was asking the entire panel, so if you want to comment on that, you can, and you don't have to.
I'm not a bioethicist, but I do think that the response from the community, the immediate response from China, the immediate response from the International Society for Stem Cell Research that you saw was absolutely appropriate.
I thank you. I thank
yield
Ms.
back. Merry Christmas.
I thank the gentleman. The chair recognizes the gentleman from Ohio, Mr. Jordan.
Thank you, Mr. Chairman. Dr. Temple, are you here today representing the International Society for Stem Cell Research or the Neural Stem Cell Foundation, or both?
Both.
Okay, and you run the Neural Stem Cell, that's the
I do.
business you run, okay. Do you guys get tax dollars? Do you get grant money from the federal government?
We do.
You know how much?
I can give you the exact, I think I submitted the exact grant information.
Okay, I have that here. And this is for the last few years?
Yes.
I do a quick, looks like over $6 million. Is that about right?
Yes, probably.
$6 million in the last 3 years? Taxpayer money? Okay,
Yes.
do you get other funding?
I do.
What's your overall budget?
It's a few million per year, three to five per year. I think I can get you all of these details.
$3 million a year is your overall budget?
I would like to make sure that that statement is correct.
Well, we know it's over
submit
6 million.
it.
We know it's over 6 million you're getting in 3 years' time, which would, you know, a little over 2 million a year from the taxpayers. I'm curious, did you happen to see the Planned Parenthood videos a few years ago that got such wide attention?
I did.
And what did you think of those?
No, I am here to answer questions about the alternatives or possible alternatives.
You don't think that's relevant?
To fetal tissue research. I know that there has been
Dr.
Temple,
you have to answer the questions.
I am answering.
Unless you're claiming some privilege, you have to answer the question.
So I found the videos shocking. Yes, indeed.
Okay.
I don't know if that is a representation of the process. I think it is not, because we know that this tissue is obtained under strict guidelines that have been established now for many years and with appropriate oversight.
Well, when the doctor from Planned Parenthood was asked if procedures are altered to obtain the kind of tissue that purchasers were seeking, she responded, if you maintain enough of a dialogue with the person who's actually doing the procedure so they understand what the end game is, there are things you can do, changes that can make their technique to increase their success. So they were actually changing procedures in order to harvest certain tissues. You agree with that?
I think that in the statute that is not allowed.
Do you guys, where do you get the fetal tissue that you use in your studies and experiments? Where do you purchase that tissue?
So you will see from my publication record that we have worked with fetal retinal pigment epithelial cells. Those cells were sent to us from the National Institutes of Health.
So you only get the material you use, you only get from NIH?
In that publication, that is correct.
Well, I'm not asking you just about that publication. I'm asking you the fetal tissue that you use in your research. Where do you get that tissue?
We have also received tissue from ABR.
So you get it from the same people who were buying the tissue from Planned Parenthood?
Yes.
Same group of folks?
Yes.
Okay.
Yes.
Okay. And do you do other research that is not fetal tissue?
Yes, I do.
Okay. Just was interested in that. And so there was, where's the grant? That one I want to look at here, the grant issue. Are you going to apply for the, recently NIH said that there's 20 million, will you be, that is alternative non-fetal tissue, are you going to be applying for some of those grant dollars?
I think it is important to explore alternatives. Alternatives certainly may be useful. The key point is that in order to know if a new model is as good as the old model, you do need to compare it to the model made with fetal tissue. So I would like to read the full RFA in order to make that determination and to see whether our research would be suited.
How long you guys been, how long you been in existence?
Oh, 11 years.
Okay. I see I'm out of time, unfortunately, Mr. Chairman. I'll yield back.
The chair recognizes himself for a series of questions. So, Dr. Sandra Lee, let me come to you. You've heard most of the testimony here today, and do you agree with Dr. Temple that if we do away with the purchase of fetal tissue, that there is no viable alternative?
I strongly disagree with that statement, with the statements that she's making, because I know from, thank you, I know from all of the literature that I've been reading, I know from my own research, I know from talking to colleagues that there are several successful alternatives available now.
Dr. Prentice, do you agree with Dr. Temple?
Mr. Chairman, my colleague and I, Dr. Sandra Lee and I, have spent a lot of time and a lot of paper documenting that there are multiple alternatives to the use of aborted fetal tissue.
All right, so Dr. Temple, obviously you disagree, and so all three of you raised your right hand and agreed to swear under oath that your testimony was the truth and nothing but the truth. So tell me, Dr. Temple, where are we at odds? Obviously, you can tell I'm at odds with you, but I'm going to try to do that in the civil manner. But where are we at odds? Because what I'm trying to find is the breakthroughs that we've had in science. Long after the study that Ms. Chu recommended to our attention, long after that, we've had breakthroughs in real science in terms of what we can do, whether it be humanized mice, whether it be using the discarded tissues from neonatal cardiac surgery. I mean, when we look at this, we've had unbelievable breakthroughs, and so I'm guessing a practice that has been typically there. Is there nothing out there that would suggest that we can use other alternatives like the two doctors to your right have suggested?
You ask what the difference is. I think it's a difference in interpretation, but it's also
interpretation
a difference
of what?
In the details and an understanding of the details. These are complicated models. They're complicated diseases. You need to really delve into the details, which is why we rely on experts in our community who are truly experts in these different fetal tissue models and in organoids and in all the alternatives that have been suggested today. And the consensus opinion is that those alternatives are not sufficient.
All right, so let me ask you this, Dr. Temple. And I think it was Dr. Prentice that talked about the fact that if you have someone doing cardiac surgery on either in vitro or not, that there is discarded tissue that would have the same properties. Do you agree that they would have the same properties as an aborted fetus?
Not at all, no.
Okay, and why would they not have the same properties for research?
Yes.
Let me clarify, for research.
Yes.
So you're saying scientifically they do not have, so we can abort a fetus and we can take that same gland and they would not have, it's the same material, but it wouldn't have the same properties.
It is not the same material.
I know
it is different developmental stage and it has unique properties.
Okay, so what developmental stage are you recommending that we, because I know that you've been purchasing, so what developmental stage do you purchase fetal tissue?
So,
I mean, from ABR, what are you purchasing from them?
So for the BLT mouse, we do not generate that mouse, but I think I can provide the information as to.
But you said you earlier when Mr. Jordan asked the question, you said that you bought fetal tissue from ABR?
Yes, we
At what period of gestation are those fetal
I
parts?
would like to provide that information later so that I make sure
Generally,
it's
what
accurate.
first trimester, second trimester, third trimester, do you buy them all?
Not to be, so it would be earlier, most likely, but I would like to provide that if I may.
So you've never bought third trimester?
No.
How about second trimester?
I would like to provide that if I may.
So you may have done second trimester.
I would like to provide that information.
Okay.
I cannot
How long would
recall
it take you to
because it's
provide
been
that?
a while. Excuse me?
How long would it take you to provide that information on what your particular
think
research?
Very soon, yeah, soon, a few days.
Is 30 days
after.
enough? Actually,
Absolutely, yes.
Okay.
Yes.
All right. I don't know that you can see this chart, but each one of you have been given a chart. And so the top part of that is actually the amount of money that is charged for fetal specimens, and that comes from Senate testimony. The bottom issue there is really the amount of money that came from a House report. And so I guess for me, why would there be a difference in the charge between baby parts at first trimester versus baby parts at second trimester? Because it looks like in January 2015, it was $550 for baby parts in first trimester and then less in the second trimester. Why would there be the difference if do you have any idea, Dr. Temple?
This is not my area of specialty, as you know. So I'm unable to answer your question.
All right. So I guess the question then, it looks like from ABR, the same place that you buy some of your fetal tissue from, they sold 80 fetal...
I should clarify that we do not purchase and have not for some time. Just so that you know.
You've not purchased what?
Any fetal tissue for some time.
All right. Well, you've been in existence for 11 years.
Yes, within that time frame we have. But
so
not
when did you stop buying it?
Again, I will get all of this detailed information for you within 30 days.
So what you're saying is the purchase of fetal tissue is not key to your research?
It's not ongoing. I am here also to represent the International Society for Stem Cell Research, the members of that society, and others in the scientific community.
I'm a little confused here. If you were using it and you stopped using it and now you're doing something else, is it just because you stopped doing the research, or?
At the present time, we are working on other stem cell types. But if there was a need in our research
fetal
to do it,
stem cell types?
Actually, no, we do.
So you've taken an alternative method that you're looking at now.
For the particular disease that we work on, which is age-related macular degeneration, also tauopathies, we've been able to do this research without access to fetal tissue, but in the past we have used it in order to benefit our research programs. And
So Dr. Temple, are you telling me that science has actually progressed where you don't have to use fetal tissue for the same research, or you're not saying that?
This is the case as we have spoken about vaccine
development. Well, that's the whole reason for this hearing. I guess the whole reason for this hearing is to not get into any issue other than, are there alternatives? And if the gentleman from Ohio offered you $20 million in a grant, to do research.
Explain?
Hold on.
I must
I'll
explain.
finish. I've let you talk all day, so let me finish. If he offered you $20 million for a new grant to do research on non-aborted fetuses, would you take that grant?
The critical thing is that it will not substitute for every application and for every disease. But scientists will use alternatives where they are available. We see the value
but they
in
haven't,
that,
Dr. Temple.
but they will not.
But that's the whole point. But they
Scientists will only use it if it's essential, if it's important, and you cannot misrepresent the science. Currently, these alternatives are not adequate.
And you're basing that based on your discussions with your colleagues?
And the consensus of the field, yes.
But you already testified earlier that you've not talked to anybody that's given you any.
No, I've spoken to many people. I just have not
you've
heard
found 100% agreement with your position.
misrepresentation of the science that I have not heard previously. Yeah.
Gentleman from Ohio.
Thank you, Mr. Chairman. I just want to make sure I understand the money again here. So there's $20 million a year that NIH is spending with experiments on fetal tissue. And I think Dr. Lee, is that right, or Dr. Prentice?
Right.
Yeah.
Correct.
But that's just what NIH is spending in-house.
Correct.
So it's actually a lot more.
Yes.
Because we know that Dr. Temple got $6 million over 3 years. Is that right, Dr. Temple?
Not for fetal tissue research.
Well, that's what I asked you, and you said you gave us this form. I said, when you add these numbers up.
That was the total. I think that's your
total.
total
Grants. That's not for
So
fetal tissue
both,
research.
Does it include fetal tissue?
I'm going to get all those details. I think one of the earlier grants. Currently, no.
Say it again. Currently, no?
Currently, no.
But over the 11 years, you've gotten a grant from NIH for some, and that would be in addition to what NIH.
That was using the fetal cell lines that came from the National Eye Institute, correct?
Okay. I just want to be clear. $20 million spent a year that NIH spends in-house, but also there's other dollars available for folks like Dr. Temple and her organization to access that also experiments with fetal tissue.
That's correct.
All right. How many other folks are there out there besides? How many folks are in that international society you're here representing today?
Close to 4,000.
So 4,000 others. So we're talking potentially, I don't know, we're pushing 100 million. Maybe it's all 100 million that I said, the bigger number. Would that be accurate, Dr. Lee?
I'm not sure I'm not sure exactly which investigator is going to, but I think it's possible that a percentage of that is going to the stem cell researchers that Dr. Temple is talking about.
So each of those 4,000, those other 3,999 could be accessing taxpayer dollars to do fetal tissue research as well.
A percentage of them then could be, yes.
Okay, so I just want to be clear. It's more money than we thought when we had our exchange an hour or so ago before we had votes, which I think taxpayers would like to know. That's a lot of their money. And again, that's in addition to what happens on the front end. Planned Parenthood gets a bunch of their money on the front end.
Right.
And then their tax dollars are also used to purchase the fetal tissue and then pay for folks who are doing research on it. Not just in-house of the 20 million, but all kinds of potentially 4,000 other entities around the country. And, as Dr. Temple just pointed out, you can use alternatives and get great results, which they're doing as we speak.
Absolutely, and scientists embrace alternatives if they are available. But we're talking today.
You've been very clear about that. I appreciate that.
The alternatives are not available for every
Dr. Temple, do you think Planned Parenthood should be getting dollars from the taxpayers on the front end?
Could you say that again, please?
Do you think the United States taxpayers should be supporting Planned Parenthood?
I do indeed.
You do?
I think, yes, they do wonderful work for women's health.
In spite of what we saw in that video, which you said you disagreed with strongly, you thought it was shocking, you still think they should receive tax dollars from constituents that I represent who find what they do to be just morally wrong? You think they should still get it?
They do wonderful work for women's health, which I think is a very, very important thing.
And if we could, to the same issue, if we could make sure whatever wonderful work Planned Parenthood does for women's health, if that is done through some other entity that gets tax dollars that doesn't provide abortion services, would you be okay with that?
The framework, the ethical framework and the oversight for the fetal tissue research is well established. The regulations are there.
I'm not talking about that now. I'm talking about Planned Parenthood, who you said earlier, when you saw their videos, you were shocked.
Yes.
You still think they should get taxpayer dollars?
I don't think that was the Planned Parenthood videos that I saw. I do believe that Planned Parenthood should get taxpayer because I believe it is an organization that benefits women's health greatly.
And what I'm asking you is sort of the same issue we're talking about here. If there are alternatives that would get taxpayer dollars that don't provide abortion services, which so many of our constituents find wrong, and alternative organizations who've never been caught on videotape saying the shocking things, to use your words, that we saw from Planned Parenthood, why not give taxpayer dollars to those organizations and not to Planned Parenthood? I mean, you said that it was shocking. You thought you were shocked by it just like Mr. Meadows and I would.
This is true. The key thing, I think, is to realize the status of the science and to understand that the researchers are doing this to create knowledge, but to create medicine. That is why we are doing this. And we will use fetal tissue if there are not alternatives. I think that we should research into alternatives, but we cannot underscore how valuable the research is now, and that in many instances it is irreplaceable.
All right, Dr. Temple, let me ask you this last question then. Do you believe it is harder to find a cure for some of the diseases that you've talked about than it is to figure out how to use non-aborted fetuses to do that research? Which is easier?
So I think that's a very astute question. In talking to somebody who is an expert on HIV/AIDS recently, they talked about the interaction of that virus with the human immune system and how complicated it is, and that we need to study that in the best possible model that we can, which is currently models like the BLT mouse. And this is ongoing research. If we were to stop this,
so let's assume I keep the same research going. I get. Yes. If you're in the middle of a clinical trial and all that, we've got. But what I'm talking about is going forward. So if we did not continue that, could the scientific community, the 4,000 people that you come up with, that you represent, could they not find an alternative out there that would provide the same clinical trials and the same research ability easier than perhaps even the disease that they're looking to cure?
So we have been trying to create a bone marrow niche for 60 years around the world. That type of niche would help to supplant the current humanized mice, the BLT mice that we use. We have failed after 60 years around the globe to recreate that bone marrow. I think that it is a tall order to imagine that we could do that swiftly.
So your testimony is, so I guess what you're saying is easier to cure HIV, cancer, and all the other things than it is to find an alternative in terms of researching on non-aborted.
I did not say that.
Well, I didn't think so,
Yeah.
but you keep avoiding the question that I asked. If I challenged you today and said, listen, we're only going to give grants, I'm not talking about existing research, but we're only going to give grants going forward on using non-aborted fetuses. So it would be discarded fetal tissue from research, maybe miscarriages, other things where you would actually get the same material to do research, but just by a different means. Do you think that the scientific community could figure a way to do research? Is it just a matter of cost?
So I'm explaining. We haven't done, in 60 years, we haven't been able to create the human bone marrow. Do we delay cures for 60 more years without any certainty of being able to do that?
We're talking over that, all right? So is there any way to research on bone marrow without an aborted fetus?
Currently, in order...
I'm just talking about any.
Of course, but will it reproduce? Will it reproduce the essential properties of the current immunized mice that we use, the human immune system mice that we use now? And the answer is no. We cannot artificially create it. just like brain organoids, are not the same as fetal brain tissue.
So Dr. Temple, let me tell you the problem that my constituents have.
Yeah.
And this is literally, you can make the arguments for or against abortion. You can take a pro-life stance or a pro-abortion stance. You can make that all day long. What my constituents have a problem with is using their tax dollars to buy baby brains and baby eyes to do research when they find it abhorrent. I mean, do you not see why that we have this issue? You know, because there's, you know, we spent $25,000 on 30 pairs of eyes, and 15 of those were past 20 weeks in gestation. I mean, you know, science is catching up. A lot of those aborted babies now are living because of what we've done with science. And so do you see the conundrum that we're in?
I do.
Okay.
I do, and I appreciate it. But I think if you could convey that the researchers respect and are enormously appreciative of mothers who choose to make this precious gift, this donation, and that we take that tissue and we use it for potential life-saving therapies.
Dr. Temple, I can tell you, there are some who will accept that. Right. There are most of my constituents who don't understand when you harvest baby parts, why that is okay. And I don't understand why you would do that. I believe that there are other alternatives. And so here's what I would ask you, since you don't agree with that. I ask each of you, all three of you, to come back to this committee. You've got some stuff to do in the next 30 days. So along with that, if you would come back to this committee with recommendations on how we could look at research done on non-aborted fetuses and what the cost associated with that could be. Because if there's a cost for us doing the moral thing, then I'm willing to help with that as well. And I know that new research methods may cost more, and that may be something that you're able to find bipartisan support. But if you could do that, is come with 3 recommendations that you have to the committee in the next 30 days. Dr. Sandra Lee, are you okay with that? Dr. Prentice? Dr. Temple? Okay. Yes. I thank you all for your testimony, and if there's no further business, this subcommittee stands adjourned.
Thank you.